So today we'll be dealing about two different types of electron microscope. So in this we'll be telling you about discussing about the transmission electron microscope, and one is the scanning electron microscope. So two different types of microscope, that is in short you call it as a SEM for scanning electron microscope, and TEM for your transmission electron microscope. So why do we use this? Like this is your general picture of transmission electron microscope. Then you have the setup of your scanning electron microscope. Okay. So what are these parts, or what is the content inside, how it is made? So that will come to see. So basically, like why do we use this transmission electron microscope or the scanning electron microscope is to generate the image of high resolution and in a magnified manner. While we are using the general microscopes, general light microscope, we see that the image formed is not very much clear, but it is like if the samples are very small, so those microscopic sample you can magnify up to 100x. Okay. But beyond that, if you need a greater magnification, if you need a greater resolution, so what you do, we place those samples in the electron microscopes. So the advantage of this electron microscope is that they develop a high resolution and high magnification image. Okay. So this is the actual setup, how it looks like, the transmission electron microscope. So if you observe the first word of each electron microscope, that is your transmission and scanning. Okay. So with that particular word, we can get an idea how the samples are being analyzed. So the basic is like in the transmission electron microscopes, the electrons will be able to transmit from the sample. That means the sample will be able to transmit the electrons within itself. Whereas in the scanning electron microscope, what will happen is the surface will be scanned throughout with the help of the electrons. Okay, so that is the basic difference, and there are a lot many differences that we'll study in the coming slides. So the basic is like in the transmission electron, the electrons are being transmitted, whereas in the scanning, the electrons are scanning all over the surface of that sample. Everybody, are you able to hear me? Is it clear? Yes, sir?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Okay. So next we come to the setup of the transmission electron microscope. So if you see, like we divide different parts of those electron microscope in basically three different parts. Okay. So if we divide the three essential parts or the systems of the electron microscope or the transmission electron microscope, so the first part will have an electron gun which produces the electron beam, and the condenser system which focuses the beam onto the object. Okay, so this is your first part. So here we can see the first part. This is your electron gun. Okay, so electron gun is basically your tungsten filament which is of V shape. Okay, so when the high voltage is passed through those tungsten filament, so what will happen is the electrons will be released. Okay, So those electrons, how they are released is by the process of thermoionic emission. Because once the current is being passed through this tungsten filament, as a result, what will happen is, because of thermoionic emission, the electrons will be released from that place. Okay, so from this gun, when the electrons are coming out, so their speed of electron will be amplified. Those electrons which are coming out of the electron gun, there will be an anode. So what that anode will do is that will just increase the speed of the electrons which are coming out of the electron gun. Fine. So that is the working of your, that is the function of the anode. So then again we have like just below the anode we have condenser lens. So the condenser lens is not like the conventional lens that we use in the light microscope. So in the light microscope, generally what we have is the lens is made up of what you say, a glass piece, a glass material. But whereas in this electron microscope, the lens that you see, the condenser lens or the objective lens, those lenses are actually made up of copper coils. Okay. So there will not be a glass thing, but a series of copper coils are there, which makes your condenser lens. So what the function of this condenser lens is, to confine or to condense whatever the electrons are coming out of this anode. So those amount of electrons will be collected and condensed in one small area, that electrons will fall on the specimen then. Okay, so once the electrons coming out of the condenser lens has been condensed or confined in one particular place, those things, those electrons will fall on the specimen. Okay, so the specimen is actually how it function is like the specimen will have the ability to transmit some of the electrons. At places the electrons will be absorbed, or it will not be able to pass, or at times the electrons will be passing throughout the specimen. So now what will happen is like whatever the electrons are passing through the specimen, so those electrons will be captured by your aperture lens, okay, or through the objective lens. So what that objective lens will do is that will capture all those electrons which are coming out of the aperture aperture lens, or you call it a objective lens. So once those electrons are coming out of or captured into the objective lens, after that it is again going to get amplified. It is going to pass through the intermediate lens and finally reaching to the projector lens. And this will, what it will do is it will amplify whatever the amount of electrons has been collected or coming onto this projector lens, that is going to amplify or finally make an image on the fluorescent screen. So as a result, what you are getting, what image that you are getting from the scanned electrons will be made on the fluorescent screen. So nowadays, like apart from the fluorescent screen, we also use CCD. Okay, so CCD, you might have heard the term in your photography. It is the same kind of your CCD, that is your charge coupled device. So what it does is it captures the image directly instead of using the fluorescent screen. This electrons, whatever is coming on the screen, If it is not the fluorescent screen, it can be a CCD screen. So that is just going to transfer the information from this CCD to the CPU, that is your CPU of the monitor, and finally it will amplify and recreate the image that is being captured on the CCD. That is ultimately in the result visible on the screen of the computer. Okay, so that was the first I discussed about the first portion. The second portion is the image producing system, consisting of the objective lens, movable specimen stage, and intermediate and projector lens through which the electron passes through the specimen to form a real highly magnified image. So up till this condenser lens and above, it was the first part. This is your second part. Okay. So now the third part is the image recording system, which converts the electron image into some form perceptible to the human eye. The image recording system usually consists of a fluorescent screen for viewing and focusing the image, and a digital camera for permanent records. Okay, so there will be a fluorescent screen. So the image can also be developed on the fluorescent screen, and apart from that, there can be a CCD or your digital camera also, which can record the image. So this is about your transmission electron microscope, and few more topics, few more theory we will add to that. That is, in normal microscope, light is used in form of photons for developing an image, whereas in TEM, that is your transmission electron microscope, it develops a 2D image. Okay, so this is important. Like in your TEM, 2D image will be developed, whereas in the SEM, 3D image will be developed. Okay, and the image developed by whether it is TEM or whether it is SEM. So both the types of electron microscope, they will develop the black and white kind of image. But here in the TEM, you will develop a 2D image, and in SEM, you will get a 3D image. Okay, so in TEM, what we get is a 2D image when high voltage, that is 80 to 300 kilowatts from the electron gun is being exposed. Okay, so in that electron gun, what is the voltage? It is about 80 to 300 kilovolt. Okay, and I told you about the shape of the filament, that is the tungsten filament in the shape of V. Okay, is passed, and because of thermoionic emission, electrons are released, which are further accelerated by anode and passes through electromagnetic lens, that is in form of copper coils, and falls on the sample. So this sample is actually kept on a mesh. So mesh is just like your what you can perceive is small filters are there, small mesh is there, of different size and of different shapes also. So according to your convenience of the sample, whatever is the sample you are analyzing, according to the sample, you can choose the size of the mesh and the shape of the mesh also. Okay, so I will just I'll show you the picture of these two things, like the copper coils and the mesh later on. Okay, so let me just continue with this thing. Transmitted electron passes through the series of electromagnetic lens and finally falls on the fluorescent screen, that is, or the captured device can be your CCD also. Okay, one more important thing is the whole setup, that is how from the place where the electrons are being released until the time the electrons are captured on the fluorescent screen. So the whole setup is inside the vacuum. Okay, and the pressure of that vacuum is 10 to the power minus 4. So in this setup, in this vacuum, the setup is like made because as you know, like the electrons are very sensitive, okay, and they are very reactive. So in case if there is no vacuum and air is there inside the compartment, so what will happen is once the electrons are being released from the electron gun, if there is a air molecule in that or air dust particles are there or a small any other foreign material is there, so electrons are going to react with that particles and they will give a small arc or a flash kind of thing. So as a result, what will happen is the quality of your image, it will be degraded. You will not get the picture very much clear. So the whole setup is made inside the vacuum to avoid such kind of disturbances or distortions. So the setup is inside the vacuum. So this is about your scanning electron microscope, the setup as you can see. So this setup is actually, when you see like as you compare, the sample is in the end. Okay. So this is where the sample is kept in case of your scanning electron microscope. Whereas if you compare like the where the sample is kept in case of what you call transmission electron microscope, the sample is kept somewhat here. Okay. So if you see the diagram, if you see this is sample, the specimen is kept just below your condenser lens. Okay. Whereas in the scanning electron microscope, your sample is kept just at the bottom. Okay. So here also the process is approximately same, because here also the source of electron is your electron gun, which is having the filament V shape, tungsten filament is there. But the thing is the voltage varies. Okay. So in TEM, what we were using is, for transmission, we were using high voltage, that is 80 to 300 kilovolt. Okay. Whereas in this electron, scanning electron microscope, we will use somewhat high voltage, but in comparison to TEM, it will be a low voltage, that is about 1 to 30 kilowatt. So if you compare the TEM, there we have been using 80 to 300 kilovolt, whereas in SEM we use 1 to 30 kilovolt. Okay, so that is the voltage difference. And in the last slide also we'll see like in detail or in crux what is the difference between the SEM and TEM. So first let us discuss about this thing, that is your scanning electron microscope. It helps in studying the surface of the solid objects that utilizes a beam of focused electrons of relatively low energy as an electron probe that is scanned in a regular manner over the specimen. So what it is doing is scanned in a regular manner over the specimen. Okay. So whatever electrons is being like focused on the sample, those electrons will like completely cover the it is going to scan throughout the sample. It is not like a small portion of the sample will be covered, because it has to make the image. So getting the scan from all the possible direction, from all the possible angles, will lead to the quality of your photograph ultimately. Okay, so the image will be clear, the image will be good only if the scanning has been complete from all the directions. Okay. the action of the electron beam stimulates emission of high energy backscattered electrons and low energy secondary electrons from the surface of the specimen. It is desirable that the specimen be electrically conducting, otherwise a sharp picture will not be obtained. Okay, so like in these last lines, we will be discussing about like the sample preparation. Okay, how the sample should be prepared. So conductivity is usually achieved by evaporating a film of metal such as gold. That is, the thickness of the gold material should be like 50 to 100 angstrom thick on the specimen in a vacuum. Such a thickness does not materially affect the resolution of the surface details. That means in the last two paragraphs, in the last few lines, two important points are to be taken care. First thing is like the sample preparation. Okay. So while we are doing the sample preparation, this is an entirely different process. It is a very complex process. Like in your electron microscopy, the main effort, the main hindrance, or you can say the main labor is while preparing the sample only. Okay. So the toughest job while using your electron microscope is while making the sample. Okay, while getting your samples ready for the electron microscopy. So in that electron microscopy, we have different steps. That is the fixation you have, then after that you have rinsing, that again, then you have secondary fixation is there, then you have your dehydration is there. So ultimately what happens is like while we are making the samples ready for the electron microscope, there is one important point to be noted, like the electron microscope, whenever we are using, this will never be used for your living cells. Okay, so living organism cannot be scanned in the electron microscope, because while you are making the sample ready, so when you are following those processes of electron microscopy, so while you are making the sample ready for the analysis, you have to do fixation, you have to do rinsing, you have to do secondary fixation, then again you have to do dehydration. So as a result, what will happen is the cell is going to dehydrate, it is going to die. Okay, so once the sample dies means ultimately it will no longer be a living cell. So it is but obvious that whatever sample we are using, it should be a non-living. Okay. So like one important thing I would mention here is like the sample preparation. Like in the last few lines, it is said that the sample should be conducting. Okay. So once the electrons are falling on the sample, if the sample is not reactive, it is not able to conduct the electrons, what will happen is you will get a complete black image, because the because whatever the electrons are falling, okay, so those electrons, whatever is falling onto the sample, it has like some possibility that either the electrons falling onto the samples, it will either be deflected, okay, or it may be reflected back, okay, or there can be a X-ray generation. Okay, so for any samples, okay, for any samples, when the electrons are hitting on the surface of the sample, okay, So within that sample, when the electrons enter, so it is possible that the coming electrons from the electron source, from the electron gun, it may enter into the sample and it may expel some of the electrons from the sample. Okay. So once the electrons coming from the electron gun hits the sample and the electron from the sample is emitted out, so that kind of electron, you call it as a secondary electron. Okay. So those kind of secondary electrons are detected by your secondary electron detector. Okay. So it should be clear, like electrons coming from the electron source, that is your electron gun. Once it hits the sample, there may be a possibility that the electrons within the sample will be released. So those released electrons will be captured or detected by your secondary electron detector. Fine. Then there may be a possibility that some of the electrons might not enter, but it may be reflected back. Okay. So that kind of electrons, that you call it as a back scattered electron. Okay. So that is one possibility. That is actually those back scattered electrons are detected by your back scattered electron detector. Okay. And there can be a possibility, like once the electrons are like hit the sample, once the electron hits the sample, the electrons may reach inside. Okay. And the sample may generate a photon. Okay. So once the electrons inside the sample reaches and it comes out in the form of photon, it is in the form of X-ray. So those kind of X-rays are detected by the X-ray detector. Okay. So these three things, you should be able to memorize and make it clear. Like once the electron falls on the sample, there are three possibilities. The three, the three types of possibility may be either the electron from the sample, the electron may either hit the sample and release the electrons. That is one possibility, which is a secondary electron and being detected by the secondary electron detector. The other possibility is either it may hit the sample surface and it may go back. So that is a kind of your reflected electron, and those are like captured by back scattered electron detector. So again, you have like the third possibility is that the electrons may enter inside the sample and it may be like converted into photon and released. That will be in the form of X-ray, and that is being detected by your X-ray detector. Okay. So the possibility you can see, and the three different types of detectors used in your scanning electron microscope. Okay. And coming to this gold plating, that is the sample, whatever sample we are using, first thing is it should be a conducting. So if it is not a conducting, so what we are doing is inside the vacuum, we'll make a very thin film of gold. The thickness will vary within 50 to 100 angstrom. Okay. So that thickness of your gold is not going to affect the resolution or the shape of your image or the sample. The sample will not be affected if we make the coating, conducting coating of 50 to 100 angstrom of gold. Fine. So that was about your scanning electron microscope. A few things to be added, like electrons coming out of the electron gun are accelerated by anode, and passes through a series of electromagnetic lenses which are directed on the sample, and the electrons deflected in form of secondary from the sample, and back scattered electrons which are reflected are captured by secondary electron detector and back scattered detector respectively. Then the third possibility was like the generation of X-rays. So those electrons captured by the sample turns into photons and being released. Those things are captured by the X-ray detector. And it is also known as your EDX, or your energy dispersive X-ray analysis. So by these three different types of detectors, whatever the data is being detected or captured by that detectors will be transferred to the CPU, and the CPU will ultimately make a resulting image from those three detectors, and you will get your analyzed image or the final image of the sample that you are viewing under the microscope. So is it like clear with these two types of microscopes? Transmission electron microscope. Here we are using the electron gun. Anode is accelerating the electrons. Then you have your condenser lens, which is condensing and focusing on the specimen. The specimen kept on the mesh. The mesh is just like it will vary within the shape and the size. And the electrons which are coming out of the specimen passes through your objective lens and intermediate lens, and finally projected through the lens on the fluorescent screen or the CCD screen, finally being transferred to the CPU, and the resulting image on the screen you will get. That is one thing. Then about the image, like the instrument, how much voltage it is using. It uses a high voltage, that is 80 to 300 kilovolt. As a result, you will get a 2D image, a 2D black and white image, and the shape of the filament is your V-shaped tungsten filament, and the whole setup is under the vacuum, that is for 10 to the power minus 4 pascals. In the scanning electron microscope, we use the same source electron gun. Then you have your electrons, then you have your condenser lens. So those condenser lens will pass through the objective lens. After that, the electrons are focused on the sample, and the whole area of the sample is scanned throughout the sample, and the electrons generated from the sample surface will be absorbed or will be detected by the backscattered detector. Then you have your secondary electron detector and X-ray detector, and the information from the detector will be passed on to the CPU. The CPU will ultimately it will make a final image on the screen of the sample. So this is the, what you say, the difference between the SEM and TEM. Okay, in the TEM you have like same high voltage through the electron gun, but here the sample you are keeping is here. Okay, there is a moving stage. So what you do is you take out the stage, you place the sample, and you keep back the stage. So that is there. Then you have here the stage is moving. Okay, so you move your stage throughout so that whatever the electrons are falling on the sample throughout, while moving the stage, the stage will move throughout the sample, and all the hitting electrons will be like the possibility of all the direction of the electrons will be captured, or it will be emitted throughout the sample. So we'll take some differences between the two different types of electrons. So the depth of resolution will be, in case of SEM, it will be 10 to 100 nanometer. In case of TEM, it will be 1 to 10 nanometer. Lateral resolution, it is 1 to 10 nanometer, and in case of TEM, it will be 0 to 1 to 1 nanometer. Magnification is 10x to 5 lakhs in case of SEM. In case of TEM, it increases from like 2000x to 10 lakh x. Operating voltage will be low in case of SEM, that is 1 to 30 kilovolt. And in case of TEM, there will be a high voltage, that is 80 to 300 kilovolt. The image will be formed, will be same like black and white for SEM and TEM. But in case of SEM, it will be 3D image. And in case of TEM, it will be a 2D image. Fine. So this is all about your SEM and TEM. And thank you for giving your valuable time. Okay.